In this session, we'll provide an introduction to the gross brain topography. We'll address some basic issues of central and peripheral nervous system, and then building on gray and white matter, begin to identify planes of orientation of the brain and brainstem, identify some of the anatomical boundaries of the lobes, identify major gyri in each lobe, the functions of each of those gyri, and then identify subdivisions of the brainstem and the cranial nerves within those regions of the brainstem. As you know, the neuron is the structural and functional unit of the nervous system. Essentially, all of the nervous system is built on the activity of neurons. You've seen these structures, but I'd like to emphasize a couple that we'll be turning to later on. And that is, in addition to the cell body and dendrites, it has a long axon and some terminals for delivering synaptic input to another neuron or to a muscle. Within the axon, it's very common that an axon is covered by myelin. In the central nervous system, that myelin is made by oligodendrocytes. Within the peripheral nervous system, there's also myelin, but that myelin is made by Schwann cells. As a result, there may be some different abnormalities that affect oligodendrocytes and Schwann cells that affect conduction within this neuron. As you know, the nervous system has two major subdivisions: the central nervous system, that includes the brain and spinal cord, and the peripheral nervous system. We'd like to recognize that although we've already addressed the spinal nerves, we'd like to recognize that the cranial nerves are those that emerge from the brain or from the brain stem. We'll be looking at those in a variety of different ways shortly. When we look in the central nervous system, we have, of course, the brain and the spinal cord, and we will divide the brain up in a variety of different ways. First of all, we have the cerebrum. You see here a gray structure, and that includes two cerebral hemispheres. And then deep to the cerebrum is the thalamus, and immediately below it, the hypothalamus. Below the thalamus is the brainstem. And the brainstem includes the midbrain, pons, and medulla. And then attached to the brainstem is the cerebellum. If we look at the base of the brain, we can see the cranial nerves emerging from the brain and the brainstem. Cranial nerve 1 is the olfactory nerve, and it comes into from the olfactory nasal mucosa into the olfactory bulb, and from the olfactory bulb to the temporal lobe. The optic nerve, cranial nerve 2, comes from the eye and comes into this region through the optic nerve, through the optic chiasm to the optic tract, and travels on back to the thalamus. And then we get into the cranial nerves that are based either coming into or going out of the brainstem. Cranial nerve 3, large cranial nerve, comes from the midbrain. Cranial nerve 4 also comes from the midbrain a little more laterally. Cranial nerve 5, one of the largest cranial nerves, comes off of the lateral pons. Cranial nerve 6, we can see here at the pontomedullary junction. And if we go a little more laterally on the pontomedullary junction, we can see cranial nerve 7 and cranial nerve 8. That is the facial and vestibulocochlear nucleus nerves. And then as we go down to the medulla, we can see cranial nerve 9, 10, and 11 clustered together. And then cranial nerve 12 is the last, here between the pyramids and the olive. If we look at the brain from the left, looking at the left hemisphere and cerebellum, all of the tissue that we're looking at is cortical gray matter made up of cell bodies, and you can see these large gyrations, these gyri, and sulci between the gyri. They're actually continuous from one gyrus to the next in general, except when we get to major fissures that separate out one major region from another, as we see here in the lateral fissure. The cerebellum also has these gyral patterns, but they're called folia. Separating the gyri on the cortex, we refer to sulci, or deeper fissures. Separating the folia on the cerebellum, we refer to as fissures. Notice that a good deal of the gray matter, that is of cell bodies, are buried within the walls of these sulci, so we don't see them from the lateral surface. If we were to look at a neuron whose cell body is sitting in the cortex and dissect out the surrounding tissue, we're looking from a lateral view, we can see here a cell body sitting up in the cortex, sending its axon all the way down through the deep white matter. White matter is deep to the cortical gray. And some of those axons will travel all the way through that white matter, right down through the brainstem on into the spinal cord. Others will travel from one area of the cortex to another. The one that you see here represented in red is a representation of a neuron in the corticospinal tract. If we take a horizontal section through the brain and stain it so that gray matter, that is cell bodies, appear purple and white matter appears white, we can see some major issues that we'd want to discuss. And that is, the outer surface is all composed of gray matter. And then deep to the gray matter, you see white matter traveling up into the gyri, just below and never reaching the gyral surface. And that's all referred to as subcortical white matter. And then also, we can see a number of deep structures that are gray matter, and these are nuclei, deep nuclei. This one is the caudate nucleus, which is part of the basal ganglia. We can also see the thalamus, a couple of nuclei within the thalamus. And we can also see the corpus callosum, a commissure between the left and right hemisphere, and we can see it here, and we can see it here, and it extends all the way at different cuts. We would see it at other levels. There are several different terms that are used to describe collections of axons known in white matter. The most prominent include pathways and tracks, collections of very large numbers of axons that generally travel a long distance. And in arising from a cell body in one location and traveling to another, usually the name identifies where the cell body of origin is and where the destination is, so that the corticospinal tract identifies a tract that travels, carries neurons that originate in the cortex and end up in the spinal cord. Other groups of fibers that travel together may be referred to as fasciculi, little bundles, funiculi, strings of axons, peduncles, that is little feet, lemnisci, that are ribbons, columns or projections. We've encountered a couple of these already. The lateral fasciculus, the cuneate fasciculus in the dorsal columns, the gracilis fasciculus, and then the lateral funiculi that included the corticospinal tract as well as the lateral spinal thalamic tract. Decussations are those axon bundles that travel some distance originating at one location on one side of the nervous system, central nervous system, and traveling over to the other side of the nervous system, but arriving at a different location. And that's the case for many sensory neurons that originate at one location in the spinal cord and will travel over to the other side to arrive at a different location at another level, perhaps in the thalamus or in the cervicomedullary border. We'll also see commissures, such as the corpus callosum, that carry information from one side to the other side to comparable locations in each side. and then generally efferent fibers are those leaving a location, and we'll describe them as efferent from that site, or afferent if they're approaching a location. For sensory systems, we will also use the term modality and break up the various sensory inputs into those that carry distinct functional properties. And that is those fibers that carry information about pain and temperature, others that carry information about touch or deep pressure, and others about position of a particular limb in a part of the body. If we were to take the brain and make a symmetrical cut down the midline, then each of those hemispheres, if we were to look at the middle, would give us a sagittal location, a sagittal plane. And an electrode, a slice off that midline, but parallel to it, would be in the parasagittal plane. Perpendicular to this sagittal perspective is the horizontal, is the frontal or coronal perspective. That frontal or coronal perspective has faces from the left to the right symmetrically, and it would cut the brain in a frontal position. Perpendicular to these two orientations is the horizontal or axial or transverse orientation, and these would cut the brain from the upper portion to the lower portion, from the superior to the inferior separation, whereas coronal sections would make a separation between rostral and more caudal regions. When we turn around to the brainstem and the spinal cord, we tend to use transverse sections, and those transverse sections may be at various angles through the brainstem. And there we're more likely to refer to either dorsal or ventral positions of location within that transection. Directions have a number of different terminology that you should recognize, and that is in the brain proper, the front of the brain would be identified as more rostral, the back of the brain would be identified as more caudal, the front being rostral or anterior, the more posterior and caudal region is in the back. As you move higher in the brain, we would refer to this as superior, and as you move lower in the brain, in a horizontal section, we refer to this as inferior. Because of a very sharp angle change in the brainstem, between the brain and the brainstem of about 80 degrees, the terminology is somewhat different in the spinal cord. So the higher levels of the spinal cord are referred to as rostral, that is cervical are more rostral, and caudal areas, the lumbar and sacral regions are more caudal. In the spinal cord also, the more posterior regions may be referred to as dorsal, and the more anterior regions may be referred to as ventral. In further assessment of orientation, it turns out that the radiology has a clinical convention for both computerized tomography, that is CT, and for magnetic resonance images that are viewed as though you're looking at the patient from the feet. You're standing at the feet and looking on, and the patient is on their back, and you're looking at the patient up from the feet. In that setting, the coronal sections, the patient's right side is on your left side, and vice versa. The same thing for horizontal sections. When you're looking at your left side, that's the patient's right side. And you can see this. So these are reversed relative to your position. Your left is the patient's right. If we look at a gross picture of the cortex, we can see that the two hemispheres are interconnected by the corpus callosum, white matter structures, that each hemisphere has 4 lobes, the frontal, parietal, occipital, and temporal lobe. The medial region, just between the callosum and these other lobes, are limbic structures that were sometimes referred to as the limbic lobe, even though they occupy part of the frontal and parietal and temporal regions. And then deep inside this lateral fissure, we'll find is another structure known as the insular lobe. The frontal lobe is separated from the parietal lobe by this central or Rolandic fissure. The frontal lobe is separated from the temporal lobe by the lateral or Sylvian fissure. If we remove the coverings of the brain and just look at the cortex, and then open up the fissure between the temporal lobe and the frontal lobe, we can see a number of other structures that have small gyri. That's the insula. Key landmarks that we want to start out with are separating the frontal lobe from the parietal lobe at the central sulcus. And you will identify the central sulcus by looking for a major sulcus that separates 2 gyri and does not come all the way down to the lateral fissure. It's about halfway back in the brain. A more prominent fissure is the Sylvian or lateral fissure, separating the temporal from the frontal lobe. It also separates the parietal lobe from the temporal lobe. If we remove the brain from the skull, we can identify the anterior cranial fossa, where the frontal lobe sits, the middle cranial fossa, where the temporal lobe sits, the posterior cranial fossa, where the cerebellum sits, and then over the cerebellum is the tentorium cerebelli, a tent that covers the cerebellum, and sitting on top of the tentorium cerebellum is the occipital lobe. Some structures that we want to identify in each of these lobes include in the frontal lobe the opercular, triangular, and orbital frontal gyri. These will turn out to be of great interest, these relative to language expression, and the orbital frontal relative to emotion in particular. We can also recognize immediately anterior to the central sulcus is the precentral gyrus, and immediately posterior is the postcentral gyrus. In front of the precentral gyrus, we will see the superior frontal, middle frontal lobes. These smaller gyri are included within the inferior frontal lobes. In the parietal cortex, in addition to the postcentral gyrus, we can see the superior parietal lobule, and then 2 gyri, the supramarginal gyrus, which is just above the lateral fissure, and the angular gyrus, which is capping the lateral fissure. And then the temporal lobe, we can identify superior temporal gyrus, middle temporal gyrus, and inferior temporal gyrus. The cerebral cortex is made up of a variety of different types of tissue. There's primary sensory areas that get information either about somatosensory information from the body, visual information, auditory information, or olfactory information. Primary motor areas that give rise to corticospinal and corticobulbar tract and are very important in movement activity and in the strength. Secondary motor areas are involved in some of the planning involved in movement and associated activities related to movement that we'll discuss. Association areas refer to regions surrounding the primary sensory areas that are involved in higher level integration of sensory information. as well as bringing together multiple modalities, that is visual and auditory information, visual and somatosensory information, so that you're able to smell, taste, and feel an apple at the same time and put them together. Limbic areas that we've talked about are involved in emotion and cognition and memory. We can take the brain in two major different roles and identify major motor functions and sensory functions that are rather distinctly mapped out on portions of the brain and identify some of the functions associated with these regions. So for primary motor cortex, we're talking about precentral gyrus, particularly the origin of corticospinal, a primary origin, corticospinal, and corticobulbar fibers. The corticobulbar fibers are those that go to cranial nerve nuclei in the brainstem. Secondary motor areas such as Broca's area, that's involved in motor speech, frontal eye fields involved in moving the eyes to the opposite direction from the hemisphere they're located in. Pre and supplementary motor areas involved in movement strategy and planning. And then prefrontal cortex, involved in what we sometimes refer to as higher functions, cognitive functions, judgment, emotions, inhibitions, and some limbic associations. A major other subcategory that is mapped over the brain are various sensory functions. Primary sensory cortex that we've already talked about, somatosensory information for position, as well as temperature, pain and temperature and vibration. And then olfactory, auditory, and visual information. Second sensory areas are primarily located in superior parietal and inferior parietal lobules, and they're involved in body image and spatial awareness and position of limbs in space. Wernicke's area is involved in language comprehension and corresponds to language production in Broca's area. And then the occipital eye fields are involved in form, vision, color, movement, and feed into the frontal eye fields in the frontal lobe in order to initiate a movement, eye movement. We can see all of those different structures and functions represented here with frontal involvement in motor activity for different parts of the body. And now we start to see that the leg movement is represented most dorsally, arm somewhat more laterally, and head most laterally in the precentral gyrus. That is, there is somatotopic representation of different parts of the body. Correspondingly, for the somatosensory information, it's also represented somatotopically, and that is the leg is most dorsal, the arm more lateral, and the head most lateral. The frontal lobe, we've identified a number of different structures in it and recognized that most of the emphasis will be on particular emphasis will be on the triangular and opercular areas that are involved in language function in the left hemisphere in most people. Important to put emphasis on the left hemisphere in most people. This is referred to as Broca's area for speech production, expressive language. And next is the precentral gyrus in the primary motor area. It is the principal outflow for these major corticospinal and corticobulbar pathways. And they're involved in the control of contralateral motor neurons in the spinal cord and in the brainstem. The precentral gyrus has a very strict somatotopy with motor homunculus that we'll look at in a moment. And then the other areas immediate anterior to the precentral gyrus are involved in modulating movements and controlling and planning for movements, and include the frontal eye fields inferiorly. The parietal lobe is involved in somatosensory function primarily, and in association of somatosensory information with other inputs, that is auditory and visual inputs. The distribution of those functions for primary motor and primary sensory information, somatosensory information, are represented by these maps Here you see a coronal section through one half of the brain, and recognize that the foot is represented on the interhemispheric fissure, the trunk on the vertex, the hand more laterally over the cortex, and the face most laterally and the tongue most laterally. And recognize that the most cortical tissue is devoted to those areas that have the finest level of motor control. Correspondingly, on the right, we see areas that have a direct appearance similar to that of the face, of the motor homunculus, and that is with the foot and leg on the interhemispheric fissure, trunk on the vertex, the hand more laterally, the face more laterally, and the tongue most laterally. All in the postcentral gyrus, and these are all in the precentral gyrus. In the temporal lobe, we have two major areas that we pay attention to, and that is in the superior temporal lobe on the dorsal surface. We will find an area that is known as the primary auditory cortex in the superior temporal gyrus. And then at the posterior portion of the superior temporal gyrus, we'll identify Wernicke's area, an area responsible for comprehension of language. In the mid-sagittal view, we can see a couple of areas of interest. Anatomically, we can see the occipital cortex involved in vision, and in particular, the calcarine cortex, seen right here, is the area of central vision. And the most posterior portion right here is involved in central vision at the midline. We see the occipital cortex separated from the parietal cortex by the parietal occipital notch. And then we see an area that's of interest, the paracentral lobule. That includes the leg and foot area for motor structure control, and the postcentral region, portion of the postcentral gyrus, for leg and foot sensation. And we can also see the thalamus, hypothalamus, and the brainstem, the midbrain, pons, and medulla. And we see at the midline also the corpus callosum and cingulate gyrus. If we look at the basal surface of the brain, we can see a number of structures we'll bring attention to. This is where the brainstem has been removed, and we've cut the brainstem off at the midbrain. And we're able to see the olfactory bulb and tract. We're able to see the optic nerve. We're able to see the infundibulum, which is the stalk that leads to the pituitary. And we're able to see the parahippocampal gyrus in the temporal lobe. And deep to the parahippocampal gyrus, buried, we cannot see it, is the hippocampus. Those areas, the parahippocampal gyrus and hippocampus, are involved in memory formation. And then just medially to the parahippocampal gyrus, we can see the uncus, a little bump on the medial surface of the temporal lobe that's involved in olfactory function. Just lateral to the uncus is the tentorial notch, and there we can see a region that is of some significance because of pressure that comes down from above the tentorium, that is in the temporal lobe, which lies above the tentorium. If it's enlarged by a hemorrhage or by a tumor, it may push against this tentorial notch, pushing the uncus into the nearby structures. The nearby structures include the third nerve, which is involved in eye movement and pupillary response, or the brainstem. And if it compresses the upper brainstem, it may result in coma, that is unconsciousness. In this section, we can see where the brain has been removed, and only at the midline do we see the falx cerebri. And then the falx cerebri leads into the tentorium cerebelli, which covers the tentorium and travels up over the temporal lobe. And right here is where we would see the uncus sitting right here. If we look at a coronal section, MRI, we can see the tentorium cerebelli separating the temporal lobe from the cerebellum and from the brainstem. The brainstem comes down through the tentorium cerebelli. And we see this tentorium over here. If we look at a blow-up of a fresh specimen of brainstem at a mid-sagittal level, we can see a number of important structures. And I draw your attention first to these areas that are involved in cerebrospinal fluid is generated in the third and the lateral ventricles, and it flows down through this aqueduct, the cerebral aqueduct of Sylvius, into the fourth ventricle. And from the fourth ventricle, it flows out to the foramen of Magendie and some other foramen out around the brain. And then on the roof of the fourth ventricle, we see the cerebella vermis. We see the tentorium cerebelli, this location, the occipital lobe just above the tentorium, and the brainstem is below, midbrain, pons, and medulla. And anterior to all of these structures would be the hypothalamus and the thalamus. Just above the cerebral aqueduct, we see the midbrain tegmentum. That's the superior and inferior colliculi that are involved in eye movements and auditory function. And then just above the midbrain tegmentum, we see the pineal gland. Now, the brainstem has these three major subdivisions, midbrain, pons, and medulla. And it functions both as a site of origin or destination of cranial nerves, as a conduit for fibers that are coming from the cerebrum down towards the spinal cord, or from the spinal cord up to the cerebrum, and for certain integrative functions. The integrative functions include cardiorespiratory control that you've recognized already, certain complex motor patterns, and then in particular, it controls certain aspects of arousal and consciousness, particularly a structure known as the reticular formation, which is the central core and which is critical for consciousness. If we look at the brainstem with some of the associated structures removed, that is, if we look at the dorsal surface of the brainstem with the cerebellum removed, we can see the superior colliculus and the inferior colliculi in the midbrain. We can also see the pineal gland sitting at the midline. And then as we come down, we can see at the bottom of the midbrain, caudal midbrain, we can see the fourth cranial nerve comes off. And we see the cerebellar peduncles that have been cut off here. And we see the fourth ventricle. If we look at the dorsal surface, we see the midbrain at this location. And we see the third cranial nerves coming off between the midbrain and the pons. And then off of the pons, we see the fifth cranial nerve coming laterally. We see the 6th, 7th, and 8th coming at the pontomedullary junction. We see 9, 10, and 11 as rootlets posterior to the olive. And then we see cranial nerve 12 coming off between the olive and the pyramids being a continuation of the corticospinal tract. If we look at this blow-up of the brainstem, we can see these areas now clearly, the superior and inferior colliculi, cerebella vermis, the tegmentum, the central portion of the midbrain, and travels down through the medulla and pons, as well as the basal pons, which is essentially attached onto the core of the midbrain. And we can see the fourth ventricle and the cerebral aqueduct. The tectal regions are involved in vision and hearing. If we look at the ventral view of the brainstem, we're able to see the cerebral peduncle, which is a continuation of the corticospinal tract coming from the cortex. Those fibers then dive into the basal pons and emerge on the same side in the pyramid. And the pyramid then travels down medially, just off the midline, until it gets down to the cervical medullary junction where axons will cross to the other side. We can also see the basal pons, this large structure containing many fibers that are going back and forth between cerebellum and various structures deep in the brain, as well as deep within here are some fibers that are coming from the corticospinal corticobulbar tracts. This is the basilar pons. If we look at a dorsal view of the brainstem, now with the cerebellum removed, we can see the major peduncles coming out of the cerebellum, the superior cerebellar peduncle, middle cerebellar peduncle, and inferior cerebellar peduncle, all white matter tracts that are traveling into or out of the cerebellum towards brainstem and higher structures, or getting their input from the spinal cerebellar pathway. We can also see the superior colliculus, the inferior colliculus, and the pineal gland. You can see the facial colliculus, a little bump representing a location of the facial nucleus. If we were to cut the brainstem in cross-section at each of its major levels, the midbrain, the pons, and the medulla, and stain white matter darkly, cell bodies as white, and then separately color the tegmental, the central portion of the brainstem, green. We see a large portion of the brainstem is considered tegmentum, and you see it here, here, and here. And then the structures that are stained are largely, stained darkly, are largely the cerebral peduncle, which includes the corticospinal, corticobulbar tracts here. Embedded within this region of the pons, in this basis pons, are fibers that are coming out towards us. They are also portions of the corticospinal tract. And then as you come into the medulla, you'll see a regathering of all of the fibers of the corticospinal tract. So all of the fibers here are largely coming from this region in the cerebral peduncle. And you also see a major structure in the medulla that is known as the inferior olivary nucleus.